In biology, a parasite is an organism that lives on or in another organism and benefits at the expense of the host. Parasite can cause harm to the host organism. Parasites can be found in various forms, which is endoparasite and ectoparasite. Parasites have evolved various strategies to exploit their host, and host organism may develop defense to resist or tolerate parasite infection. So the objective of the lab is first is to determine numbers of parasites in aquatic animal sample. Second is to observe any clinical sign caused by parasite on sample.
Material that we use is dissecting set, sample of freshwater and marine matter, petri dish, slide water, sardine fish in can, and microscope. Firstly, at the dorsal fin part of sample using forceps. Take the mucus at the dorsal fin and part and place in the slide. Next, see under microscope using 4T magnification and record the parasite that found and find another parasite at internal if fish like gill.
The result shows the species parasite was found is Heliplatus species and marine water species, which is Nematodes species.
Next, the organism that we found in Trichopodes spectralis, which is Eustronglides species from kingdom Animalia and family Dioctopimatidae. Last, from canine product, we found Anisakis species, also from kingdom Animalia and family Anisakidae.
In Nematode species was found parasite at skin and gills, Heliplectus species. In the aquatic environment, Heliplectus species move and eat on their own as free living organism. They live at the bottom and interact with other creatures to affect the general ecology. In Trichopodes pectoralis was found parasite at Eustronglides species. Eustronglides typically refer to parasite nematode worms, potentially dangerous for mammals' health. In can, sardine was found parasite at fish mussel, Anisakis species. Anisakis species is a genus of parasitic nematode worms, and they are known to infect marine organisms. These parasites have a complex life cycle that involves multiple hosts.
In conclusion, the parasite provided valuable insight into the diversity, characteristic, and ecological roles of various parasites. Through microscopic examination and analysis, able to identify different parasite species. This hands-on experience enhanced our understanding of parasitism in both biological and environmental contexts. The lab underscored the importance of proper techniques in specimen preparation, microscopy, and identification, emphasizing significance of accuracy in diagnosing and studying parasites.
In introduction, in mycology, Lafenol Cotton Blue staining is a magician trick for exploring the hidden world of fungi. This clever solution both preserve fungal structure on a slide and paint them a vibrant blue, making them stand out under the microscope. Besides that, this allows for detailed examination for fungal morphology, from dissecting the intricate detail of hyphae, spore for species identification, to spotting hidden fungal culprits in clinical samples.
The objective is to determine the structure of fungus using microscope.
So the material that has been used in this lab is mycelium, glass slide, forceps, toothpick, dropper, and the last one is lactophenol cotton blue.
The results show the structure macrofungus and microfungus.
PCV staining, a cornerstone in mycology, shines upon the hidden world of fungi, transforming microscopic specimen into vibrant blue landscape. This tool-active marvel both preserves delicate structures and paints them in stark contrast, unlocking a treasure trove of information. from dissecting the incisive cases of hyphae and spores for species identification to revealing lucky fungal folds in clinical samples. LPCV fuels our exploration of fungal morphology, classification, and even diagnosis, making it an indispensable tool for delving into the captivating world of fungi under the microscope.
To sum up, the terphenol cotton blue staining enables mycologists to identify and understand fungal species morphology and disease through vivid blue light, making it a cornerstone of mycology.
White spot syndrome virus (WSS) is a contagious lethal infection affecting crustaceans like prawns, lobster, and crabs, causing rapid mortality and potentially destroying entire shrimp farms. Nervous necrosis virus (NNV) or betanoda-virus is a harmful RNA virus that infects fish, causing VNN and encephalopathy with 100% mortality rates, necessitating infection control strategies for aquaculture sustainability.
For the objective, to diagnose and prevent the spread of this highly lethal and contagious viral infection that affect crustaceans and fish, respectively.
So, the result of the sample of rapid test for white spot syndrome virus is negative. Besides that, the result of nervous necrosis virus in shrimp or fish sample also negative.
For discussion, rapid test for white spot syndrome virus and nervous necrosis virus in shrimp or fish sample show no present, typically without a coloured or thin line in the test cassette.
In conclusion, the negative results in rapid tests indicate the absence of WSS and NMV in fish or shrimp samples, demonstrating compliance with biosecurity measures and best practice in aquaculture and necessitating in further research. Thank you. That's all from us. Thank you.
Gram staining is a differential staining technique used to classify bacteria as either gram-positive or gram-negative, according to the characteristic of their cell walls, both chemically and physically. Such that are gram-positive with stain purple, whereas those that are gram-negative with stain red to pink. The procedure required various types of equipment and reagents, including crystal violet, iodine, alcohol, and sulfonine.
The objective is to determine the difference between the two types of bacteria, which is gram-positive and gram-negative, according to the properties of their cell wall.
The results show for E. coli is pink while Streptococcus is purple.
The unique cell wall of E. coli, a gram-negative bacterium, is made of an ATHIRAH member containing lipopolysaccharides and a thin peptidoglycan, which give the bacteria its paint-staining ability and pathogenicity, while Streptococcus is gram-positive bacteria have a thinner peptidoglycan cell wall, allowing them to retain crystal violet dyes, resulting in a purple appearance during gram staining.
In conclusion, because gram staining and make bacterial identification quick, it is an essential diagnostic tool in microbiology. Using this method, bacteria are classified as gram-positive or gram-negative depending on the component of their cell walls. That's all from us. Thank you.